Hi, it's Meredith. I'm a dietitian. Today, I want to talk to you guys about hyperoxaluria. This hits home because in our family, we have a few people struggling with this, and it's bugged me so much because no matter how much B6 is given, they still make oxalate. And we eat a low oxalate diet. So I'm like, what the heck is going on? And I think finally I can explain it. I do think it's possible that anhydroretinol could be involved, but I also think that this process could be happening for other reasons. I'm not a person who's going to blame everything on a nutrient, although I'm really studying this one quite a bit. But it's possible that you could be making too many oxalate in your liver, and you can also make it in your kidney. You could be making too much of it because of sleep apnea or gut dysbiosis, or it could just be because you're stuck in an immune pathway called the Itaconate pathway. So I'm going to talk about that and also this hypoxia inducible factor 1 alpha, which is what happens during sleep apnea, but also during an immune response. Oh, and this isn't medical advice. It's just me talking to you about what I've learned. Please consult with your own personal healthcare provider before you make any changes to your diet, your supplements, your nutrition. That's the same thing as diet, your medications, and your lifestyle. So, this is a diagram that I made for a blog I wrote for Hormones Matter for Dr. Chandler Mars about how I think that my daughter Zoe developed vitamin A toxicity or hypervitaminosis A. So, she actually started to have low potassium. And so I decided to go on a hunt for a high potassium flour to be able to make her low oxalate bread. And I found a handout by a dietitian, and it said that plantains had 1 milligram of oxalate, sort of like a banana. And I was super excited. So I bought plantain flour and I started mixing it into her bread. And this is hindsight, but over the course of 2 years since having started that, my daughter started to walk like a drunk lady and she went downhill neurologically. And so she was on a low oxalate diet, and I didn't think anything, like, I was like, we haven't changed anything. She's not eating more oxalate. It culminated in an ER visit where she was vomiting. And they said she just has cyclic vomiting syndrome, and she was released from the ER, and they populated all of the testing to her MyChart account, and she had massive crystals in her urine and debris in her bladder. And it wasn't a UTI, it was oxalate. And it was the plantain flour because that week I had given her plantain muffins, and she loves muffins. And she was so low on energy and looked like she was lethargic that I let her have 2 of them a day. And so roughly she was consuming, oh my goodness, well, one cup of plantain flour has 525 milligrams of oxalate. So pretty large amount, because I used 2 cups in the recipe and she ate the whole recipe within a 4-day period. It was a nice giant puddle of vomit. So anyway, oxalate inhibits lactate dehydrogenase, which is a pivotal place in the cytosol where we recycle our NAD. So anytime we make lactate, we actually make some NAD. I know these arrows are pointing down, but that means that she was making less NAD. Because she was making less NAD, she could not metabolize her retinol. Here's the cofactor to go this direction. Her retinol to retinol aldehyde. So her retinol started to accumulate, and I assumed that she was stuffing it back in her liver because her serum vitamin A was creeping up. And that's a sign of what we think is a sign of excess stores in the liver. And so, I decided, when I figured out this was the cause, I decided to work on her vitamin A metabolism. And I tried trying to maximize NAD, and actually removing the oxalate helped significantly with her energy levels. We were normally on a low oxalate diet anyway. And so removing the plantain flour was like a total pick-me-up for her. She suddenly had energy and she stopped walking like she was drunk. But after about 2 weeks, all of that came back. So it was quite interesting. We had actually cut out a few other things, like we had started to worry about beta carotene because wondering if she has a slow beta carotene monooxygenase enzyme. So she had never taken a vitamin A supplement in her entire life, never. So that wasn't an issue. She rarely had dairy anyway because we were mostly dairy free. So it was very interesting that her vitamin A levels seemed to have accumulated. Anyway, if we were worried about the beta carotene because when I was researching, I found that if you are making retinol aldehyde and you're not metabolizing it to retinoic acid, then the retinol aldehyde can combine with ethanolamine and make an A2E lipofuscin, which can cause damage to mitochondria and macular degeneration and neurological decline. And Zoe, on her plasma amino acid test, always has low ethanolamine. And I was like, this is it. This is it. She is neurologically impaired because she has too much A2E because she's toxic from beta carotene. And she's not metabolizing her retinol and it's storing in her liver and it's leaking into her blood. And that was the mission. And then what I was doing wasn't helping her get better. She actually got worse from restricting beta carotene. Now, I'm not saying that we need to eat 300% of the daily value of vitamin A and beta carotene, not at all. But just trying to fix NAD levels didn't work. So, something important to know though is that retinol needs NAD, but so does alcohol metabolism. And something that is really important to understand is that inside your gut, you actually make NAD in a pathway called the kynurenine pathway. And that is highly dependent on B6, and then you also require B2. And because Zoe had been stuck in this cycle of high serum retinol for a while, that actually, retinol binding protein 4, when it's high, will actually trigger a high nitric oxide state, and that can cause a functional B6 deficiency. And so she was not using her B6 right in her body, and so she wasn't making her own de novo NAD. So her ability to metabolize alcohol was pretty much knocked out. And so that was a huge component to her downward spiral. She just, even after a few weeks of going low oxalate, she was back to the same issue. And so we're like, what is going on? So her oxalate level in her urine went down for a spot check, which we all know in the TLO group when I was in there, when I was allowed to be in there. They actually are really good about this, guys. So visit that group if you want to know more information about like dietary oxalate. They have like a spreadsheet. They're pretty thorough, so check them out. So Zoe, we went to genetics to figure out if she had a genetic predisposition for hyperoxaluria. She did not. She, when we started to supplement B6, it actually made everything worse. So that was shocking, and I'll talk about that in a second. So I have a hypothesis for you all. I have a hypothesis that anhydroretinol can cause hyperoxaluria by two mechanisms. The first one is alterations of pyruvate dehydrogenase complex activity. Either if anhydroretinol is in there, it causes a problem, or if you have low levels of retinol in the cell, it can cause a problem. And then damage to the immune function because of making anhydroretinol will also cause problems with making oxalate because it activates the itaconate pathway and hypoxia inducible factor 1 alpha. So this will be really fun. So let's look at this pathway. So this is the peroxisome, and inside the peroxisome, there's an enzyme. It's alanine. It's named after this amino acid, alanine, and the G is glyoxylate transferase, and it requires B6. And so one thing people are concerned about when they have high oxalates is they're like, I'm concerned that I'm going to turn my glycine into glyoxylate. It's going to come back through and it's not going to be able to go through AGT to become glycine again. It's just going to become oxalate. So that's a big concern, and it is possible that that could be happening. You normally would actually convert your glycine to serine in a process that I actually went over in my plasmalogen video, but it requires B6 as well. So if you add in B6, then you should be going from glycine to serine instead of going from glycine to glyoxylate, and it should solve the problem. But what we're seeing is it doesn't always solve the problem. There are people who are outliers that are still getting sick. So, I forgot to put that little label there. What about B6? So, what I think is actually happening is the people who aren't responding to B6, it's because B6 is helpful for them to make oxalate. Because what they have going on is a block at pyruvate dehydrogenase complex, or they're making more pyruvate from the itaconate pathway, or they could be making a lot of pyruvate from beta alanine as well. So it's possible that pyruvate is the issue in hyperoxaluria, and I'm going to prove that out. So pyruvate will combine with serine to produce hydroxypyruvate, and hydroxypyruvate becomes D-glycerate and L-glycerate. And the enzyme here is GRHPR, and it's a moonlighting enzyme. It also metabolizes glyoxylate to glycolate. So if you're busy over here because you have excess amount of pyruvate, and this pyruvate could even come from alanine aminotransferase. So that's another spot that I'll show you in a second. But if you're busy over here dealing with hydroxypyruvate, then you're not going to be metabolizing your glyoxylate to glycolate, and instead it's going to become oxalate. So in theory, that should stop the whole process, because oxalate is actually going to cause feedback inhibition of lactate dehydrogenase, and we should be done. There should be no oxalate production left, but that's not happening, and it's because of this. Hypoxia-inducible factor 1 alpha upregulates lactate dehydrogenase. And so no matter how much oxalate a person is producing, if they're producing this HIF1 alpha, then they're going to have extra lactate dehydrogenase, and it's not going to cause feedback inhibition, and glyoxylate will continue to be pushed to oxalate. So what about vitamin B6? Is that going to be helpful for someone who has hyperoxaluria? Well, it seems like it's not helpful. So this is a summary of longitudinal studies. Like, they did a longitudinal prospective study and looked at a bunch of things. So they looked at 7 patients with idiopathic hyperoxaluria and 10 healthy controls. And they administered 200 milligrams per day of pyridoxine, and it had no effect on urine oxalate in the people who had idiopathic hyperoxaluria. But in the people who were healthy, it actually increased their urine oxalate levels. So that's pretty interesting, right? That's a paradoxical effect. And then another 7-day course of vitamin B6 at a dose of 300 milligrams per day on urinary excretion of oxalate among 55 idiopathic calcium stone formers, and 40 with and 15 without hyperoxaluria, and 50 healthy subjects, they didn't see any changes in the urine oxalate. So B6 was doing nothing. So potentially, what's going on here? Well, what if the variability in the benefits of B6, or the lack of benefits of B6, are due to the fact that it's high pyruvate levels that it is the cause of hyperoxaluria, and people who are making oxalates but don't have the genetics to cause the problem? So the B6 basically restores the AGT function, but it's the same enzyme that's involved in pyruvate to hydroxypyruvate reaction. So you're not really solving the problem. The problem is excess pyruvate. Okay, Oh, pyruvate goes up. I forgot to click the button. I always do that. So could anhydroretinol or lack of retinol, so people who are following a very low vitamin A diet because they might think that it's a poison, could it be causing altered pyruvate dehydrogenase complex activity or function? And I got the letters switched up there. So it's PDHC, not PHCC. Whoops. Okay. So, so I think it's potentially a problem. So this is Dr. Hammerling's study on quantum chemistry rules retinoid biology, and he's finding out that retinol is required for a signalosome called PKCdelta signalosome for it to have activity. And what this signalosome does is it senses that how much energy is being produced in the mitochondria, and it will turn on and turn off pyruvate dehydrogenase complex. So we want a normal activity of PDHC. We don't want it too fast. We don't want it too slow. You have to have retinol in it for it to work. If there's no retinol, I'm sure that it's just not going to work, right? And that you'll be working around it. And that means that when you have a glucose to pyruvate, you got to put your pyruvate somewhere. And then anhydroretinol, it could overactivate PKCdelta signalosome, and then that results in overactivation of PDHC, and it could cause depletion of cofactors and eventually burnout or feedback inhibition by acetyl-CoA. And then the third possibility is a mixture of anhydroretinol and retinol, and that would actually just kind of give your mitochondria apparently a energy boost, is what the study is hypothesizing or saying, finding. Okay. One more possible thing to consider is that if you have a slow beta-carotene monooxygenase enzyme, you could be making apocarotinol, which can act like anhydroretinol in the PKCdelta signalosome, and it could overactivate it, and then that could cause oxidative stress, and it also could eventually lead to loss of cofactors and loss of PDHC function. So this is from a previous video. Are we making anhydroretinol in our body? Yes, we are. It's been shown to be made in lymphocytes. So we can do it. We can do it in our cells. All we need is a primary alcohol, a normal human body temperature, and acetic acid or some other acid. Acetic acid is strong enough to be able to do this reaction. Sulfuric acid would do it too, but we don't have sulfuric acid in our body. Hydrochloric acid would. So if you have candida in your stomach when you eat your, if you have like over-acidic stomach, you could be producing anhydroretinol. All right. So, in the absence of alcohol, we can actually convert anhydroretinol back to rehydroretinol. It's just a really crappy version of vitamin A. So more on that in a bit. So if you're not familiar with my videos, this is from the original All Roads Lead to Anhydroretinol, or actually I think this might be from the mitochondrial madness video. But basically it shows that here's the PKCdelta signalosome, here's where retinol should be. If you have anhydroretinol in there, then you have overactivation of PDHC, and it eventually leads to a bunch of oxidative stress that I show in the other video. And you could be losing thiamine, lipoic acid, pantothenic acid, or you could have eventually a backlog of the TCA cycle leading to buildup of acetyl-CoA, which would inhibit pyruvate dehydrogenase complex as well. So that's my original anhydroretinol theory of causing massive problems in the body. Okay, so this is another way to look at the TCA cycle, and you can pause the video and take a screenshot. And this is helpful for anyone who's trying to suss out, for lack of better terms, or trying to figure out where you're broken. So it shows the different inhibitors of the different enzymes in the TCA cycle. And I'm going to let you guys look at that. You can ask questions in the comment section if you want. But our issue here is a buildup of pyruvate. And it could be from multiple changes in the TCA cycle. So oxalate alone will inhibit pyruvate carboxylase. And so if you're actually a maker of oxalate or you're consuming oxalate, it can inhibit this enzyme and lead to high pyruvate. And then citrate synthase is inhibited by citrate. So if you have a block in a conatase in a buildup of citrate, then that can actually inhibit your ability to for acetyl-CoA to enter the TCA cycle, and then acetyl-CoA will inhibit pyruvate dehydrogenase, and you'll have a buildup of pyruvate. So those are the biggest ways. So if your aconitase is inhibited, that could be from overactive pyruvate dehydrogenase complex and excessive reactive oxygen species such as hydrogen peroxide or superoxide. All right, so I'm going to go on here. Here is another way to look at this. Here's pyruvate, and here are all the different ways that it can go. Again, anhydroretinol would overactivate pyruvate dehydrogenase complex, and that would lead to thiamine deficiency, resulting in shuttling of pyruvate to alternative pathways. Or lack of retinol from a very low vitamin A diet could lead to no retinol in the PKCdelta signalosome, resulting in potentially diversion of pyruvate to other pathways too. So interestingly, this first pathway we talked about a little bit, lactate dehydrogenase. This enzyme is upregulated whenever hypoxia inducible factor 1 alpha is upregulated, which happens either when you have sleep apnea, or it can happen when you when you're stuck in an immune response, the itaconate pathway. So it should be inhibited by oxalate, but it seems to not be inhibited by oxalate in specific situations. And then pyruvate can also be shuttled towards alanine, and the enzyme is alanine aminotransferase, or ALT. And this is the most common increase in labs of people who have high serum vitamin A. So that's pretty interesting. Often we think that it's liver failure, but I honestly think that it's a block at pyruvate dehydrogenase complex, whether it's lack of vitamin A inside of a cell, and I'll explain that in a little bit, or it's anhydroretinol is actually causing a burnout at PDHC, and it results in pyruvate dehydrogenase. And results in a buildup of pyruvate. Okay, so another place that pyruvate can go is to oxaloacetate, back and forth. So that's a potential shuttling back and forth. And then pyruvate can be converted to malate as well. And then one more place, pyruvate can go to the peroxisome like we discussed. So if you're trying to figure out where the heck your pyruvate is going, these are all the different places it can go. Oh, it can also go to acetate. And I have a video that I want to share with you all soon, and it's about how an overactive PDHC can cause ketosis. So you can actually turn pyruvate into ketones when your pyruvate dehydrogenase complex is overactivated. And that would be a sign of anhydroretinol toxicity, I think, because it would be normal glucose levels, presence of ketones, despite eating carbohydrates. So other nutrients that are related to a classic block of pyruvate dehydrogenase complex would be iron deficiency, because the enzyme aconitase requires iron. It could be thiamine lipoate or pantothenic acid deficiency. It could be B2 deficiency, because if you have a block at succinate dehydrogenase, and that requires FAD, then you would end up with a lot of succinate, and succinate could actually be at it's called succinylation, and it's where you put succinate on pyruvate dehydrogenase complex and it turns it off. Pretty interesting. So let's look at some lab markers. So this is one of my friends, and you can see here is her oxalate. It's very high. Her glycolate is kind of on the low end of the normal range. and her glyceric acid is elevated. So, so remember that hydroxypyruvate is converted to glycerate, and that's where that's coming from. So her pyruvate is elevated and her oxalate is elevated. So she's pushing her pyruvate into the peroxisome and combining it with serine and making hydroxypyruvate, which is metabolized to glyceric acid or glycerate. And that's actually causing competition of that enzyme GRHPR. And then she's making oxalate. So, okay. And here is Zoe. And I wrote, look at where, because I was so confused. This was when she was 2 years old. And when she was 2 years old, she had severe sleep apnea. She would have an hypoxic episode every minute, and her SATs went to 70. And we did not get that poor, sweet child CPAP until she was 5 years old because we couldn't get people to listen to us. So she was hypoxic for quite a long time. And so you can see that she has insanely high pyruvate levels, and her lactate is also, it's mid-range. So she's struggling going from pyruvate to lactate. Her pyruvate's being shuttled into the peroxisome. It's combining with serine and making hydroxypyruvate, which makes glyceric acid or glycerate. And then because of the competition, her glycolate is actually very high. The glycolate I'm going to show later is coming from the hypoxia. And so she basically is making a lot oxalate. You can also see here that her succinate levels are super high. And this is because she is also stuck in an immune pathway because she has insane gut issues. So I'll show more of that in a minute. I just wanted you to see the trends with high pyruvate will definitely cause high oxalate levels. So what do you do if the PDHC is the only issue? If it's not what I was saying with Zoe, an immune thing or hypoxia, what do you do? I think, obviously talk with your own healthcare provider, but I think that a low oxalate diet would be beneficial. Because I didn't finish that sentence, because it would help with decreasing the total burden, and it would prevent the inhibition of lactate dehydrogenase in the cytosol. And so that would allow for more of the pyruvate to become lactate. So if it's just a classic PDHC problem, that should be quite helpful. A low carbohydrate diet, let me go to the next slide. I left myself a note. This is one of my friends that I'm helping, and this is her oat while she was eating carbohydrates. I'm going to do a whole case study on her because she's quite interesting. Her pyruvate, you can see, is actually on the low end. And again, this is a moment in time. So, but she actually is doing something quite interesting. She's doing like pyruvate to alanine, and then on top of that, she has like an increased uracil, and she's making beta alanine, and she's just pushing everything to malonyl CoA. So yeah, very interesting. Anyways, so here's her eating carbs. Okay, she's eating carbs. So she's going from glucose to pyruvate, and then her pyruvate is being pushed into the peroxisome, but her lactate is also high. So it was a little bit confusing, but she has the high oxalate, she has the high glycolic acid, and she has the high glyceric acid. They're like crazy high. So let's see what happens when she decided to go keto before her next oat. And actually, she was only keto for 3 days before her oat because she kept cheating. Love her so much, because I'm a cheater too when it comes to dieting. I can't diet at all, guys. I just have to eat regular or else I go crazy. So she cheated, and so she wasn't low carb for very long. So it was 3 days on a keto diet, and her oxalate went down. So it could be a therapeutic situation. I'm not saying that that would be the permanent fix. You have to find out the reason why someone is broken. And her brokenness is melanation. She's melanating her pyruvate dehydrogenase complex because her melanate's high. And so, but it might be a temporary fix, especially if you're seriously struggling with oxalate crystals in your urine, or pain in your joints, until you can figure out what the problem is. So then you want to find what is slowing your PDHC and fix it. Is it thiamine, coenzyme A? Arsenic could stop your PDHC. FAD could be a problem, or riboflavin deficiency, because that would cause high succinate and that would cause succinylation. And then, you could have problems with malinylation. So you could have really high malinate levels. That could come from uracil. On the oat, it would come from that pathway. And that's what I'm finding that is actually a problem with methylation or a low choline intake. So you could consider that retinol is actually essential for PDHC function. So if you happen to be on a very low vitamin A diet, it's possible that you're vitamin A deficient. It is a possible thing. We don't all have max stores of vitamin A in our liver, and there's also a process by which you get retinol into a cell that could be damaged in people as well. We'll talk about that in a minute. And then you want to maximize your alcohol metabolism if you think that anhydroretinol is possible. And again, if you have ketosis and you're eating carbs, then you might be a maker of anhydroretinol. So the things that are surrounding alcohol metabolism, I'm going to touch on in a future video. Salt 1A1, it's a sulfur transferase. It requires sulfate, and people are struggling with making sulfate. And that actually has to do with stealing of the enzyme, the cofactor MOCO for the enzyme AOX. If you happen to be struggling with vitamin A metabolism, AOX doesn't require NAD. And so there's what my friend Jenny Jones, PhD, says is a MOCO steal. I really like how she says that. And then you want to try to work on NAD, because you want to get to lactate, which provides NAD for the cells. You want to get to lactate. So you don't want to have an inhibition in lactate dehydrogenase. And so a low oxalate diet would be helpful, but you have to watch out for mineral deficiencies. And then vitamin E, you need some, but you don't want it too high. So with my own daughter, and this is not a recommendation for you, but with my own daughter, she doesn't eat sources of vitamin E. She actually requires a very low fat diet because she has a decreased expression of very long chain fatty acyl CoA dehydrogenase. So she does better on a low fat diet. So we don't give her a lot of vitamin E. So I give her a supplement, and I give her 67 milligrams. I don't give her massive amounts, although that's technically sort of massive. But I think the vitamin E was key to her being better at alcohol metabolism in her gut. And then work on gut health. It's important. It's important to not have candida overgrowth or staph. Staphylococcus will also produce alcohol, I believe. I have a list somewhere, and I'll do it when I do the video on alcohol metabolism and sulfite oxidase and stuff. I'll list out the pathogens that can be making alcohol. Okay, but hold up, because there's way the heck more than this. And I know I've already been talking for 30 minutes. And also, I'm not healthy right now. So I'll probably pause the video and then come back into it for if I need to. But we got to hold on, because there's more going on than this. This is it, though. This is the reason that my sweet daughter makes oxalate in her body. It is definitely pyruvate. It's a block at PDHC. But why, right? Why is what we need to know. So we have our vitamin A related inhibition of PDHC. We've already gone over that. It's either AR overactivates it, or there's lack of retinol. Those are the two main things that I think happen that can cause a block at PDHC from a vitamin A standpoint. But there's other inhibitors of pyruvate dehydrogenase complex. There's glutarylation, and that would be controlled by hypoxia induced factor 1 alpha, which I'm going to do a whole video on these particular pathways, by the way, if I have time. There's melanation that can be from iron deficiency causing excess citrate. It could be from beta alanine that's coming from a block in urea cycle. So if you're on high dose zinc inhibits the urea cycle. It will block it. So then you'll be pushing more of your ammonia towards orotate and uracil. And if you also at the same time are not consuming choline, or if you have a slow PMT enzyme that helps you to access choline from your cell membranes, then you will have a backup of that pathway. And I need to do a whole video on that, but you'll have a backup of that pathway and it'll lead to overproduction of uracil. And then that uracil becomes beta alanine, and then that beta alanine can either become toxic to you or it can be turned into malonate, and it can malonate your pyruvate dehydrogenase complex and turn it off. And then you could be producing oxalate. It could be arsenic toxicity. Arsenic will inhibit pyruvate dehydrogenase complex. You need to have good methylation abilities to get rid of arsenic. It could be succinylation, and that is from the itaconate pathway or from a B2 deficiency. So should you go low A? That's the big question here. People ask me, like, if you think anhydroretinol is a problem, then maybe the low vitamin A diet's the best way to go. And I decided to think about it a bit more because my initial feeling on it is no, because it doesn't go along with the research of the benefits of vitamin A, but I have to suss stuff out because I need to make the best choice for my own family and for the people that I help. So let's talk about vitamin A in the gut and what it does. So this is the mucosa of the intestinal tract, and these are some cells along the intestinal tract or along the mucosa. And then there's this space in between that's connective tissue, and it's called the lamina propria. And it's just like a space that doesn't have cells except for immune cells. It's like an immune cell reservoir. And then there's a spot called the Peyer's patch. And so we actually have mucosal dendritic cells, and they can be there from there's, like, I read one study and it was like you have them when you're born, but most of them are migrating from bone marrow. So when you're born, you have some that originate there, but then your bone marrow in your bones makes all of your red blood cells and your white blood cells. And so eventually you have these mucosal dendritic cells, and mucosal dendritic cells will get retinoic acid from the epithelial cells producing it. And so this is where you want your epithelial cells of your intestine, called enterocytes, you want them to be making retinoic acid. You do not want them to be making anhydroretinol. So you want to control your alcohol in your gut. Okay, so if you're an alcoholic, there's a problem because you're probably making anhydroretinol and you're not making retinoic acid. And alcoholics don't have vitamin A in their liver. It's not stored in there. And they're like, we don't know why. Well, it's because you're making all your vitamin A into anhydroretinol and you're not storing that in the liver. It's not able to be stored because it doesn't have the functional hydroxyl group that is needed for storage. Okay, so anyway, so your immature mucosal dendritic cells, they use the retinoic acid and it changes them into CD103 dendritic cells that express alcohol dehydrogenase. So at first, they can't make their own retinoic acid, but once they've been differentiated into these mature DC cells, they can make their own retinoic acid. And these produce gut homing receptors, alpha 4b7 and CCR9, and that attracts more immune cells to the gut. So then you would get immune cells like neutrophils and other T cells and B cells coming in to help if there's like an invasion. Okay. And then these mature dendritic cells will pass on their retinoic acid to B cells, because B cells and T cells don't produce their own retinoic acid. They're reliant on dendritic cells to produce retinoic acid. And then the B cells become specialized and become plasma cells, but they require retinoic acid to become these plasma cells that make secretory IgA. And I know there are so many people out there that they do stool tests. I did one for Zoe, and I am so excited because her secretory IgA is normal. I love that. She's had low IgA for years, and it's normal because we finally figured out that alcohol was the problem. She's like an alcoholic by accident. So it's lovely. Anyway, so plasma cells make this secretory IgA, and secretory IgA will bind to LPS and inactivate it, and you'll lose it in the gut. And then also it will tag the bacteria, and it will hold on to the bacteria and prevent it from entering into the mucosa. And it tags them as well. And if they were to enter, into the mucosa or anything, then they're tagged and they can be killed easier by other T cells, I think. Sorry. Okay. So the other thing you need to know is that if there is retinoic acid present in this lamina propria from the mature dendritic cell, if the dendritic cell is making retinoic acid and passing it off to the T cells will become Treg cells. And Treg cells are what also helps to produce gut homing and immune tolerance, so that you have a good ability to tolerate and grow healthy bacteria in your gut. But if you don't have retinoic acid, then you'll make these other cells called Th7 cells, and they're more inflammatory, and they're also associated with autoimmune disease. So, what about macrophages? Because these cells right here are not really your very first line of defense. Your first line of defense is considered to be a macrophage. They're like the police officers involved in, basically walking around looking for bad guys. Well, macrophages also require retinoic acid to induce a receptor called dectin-1 receptor. And dectin-1 receptor is turned on by bacterial antigens. You can see them coming in here. And that actually makes the macrophage produce interferon, I think it's gamma, and that would attract natural killer cells to help kill off the bacteria. And so this retinoic acid is necessary for the switch in a macrophage to be turned on to become inflammatory and attract more help to the cell. And if you don't have this switch, then the macrophage just stays in a natural anti-inflammatory state. And so you won't be able to fight off pathogens. This is so crazy cool. I'm just really excited about it. So that's all really interesting, right? So vitamin A is actually needed in the gut. But no problem, right? I have stored vitamin A in my liver. That's what everyone will say to me. I've stored vitamin A in my liver. I can get it out of my liver and use it in my intestinal tract. Okay, yeah. So let's talk about that. So you get stored vitamin A out of your liver by carrying it on retinol binding protein 4. So what else is carried on retinol binding protein 4? This is from Retroretinoids and Regulated Cell Growth and Death. And it's showing which versions of vitamin A can ride on RBP4. And this is cellular retinol binding protein. It's where you hold on to your vitamin A inside your cell. And then this one is cellular retinoic acid binding protein. And obviously you can't bind retinol or this other version that's a healthy version of retinol called 14-HRR or anhydroretinol on something that only binds retinoic acid. You can bind all of them with cellular retinol binding protein. So anhydroretinol can be taking the place in your cells of retinol. And then retinol and anhydroretinol and 14-HRR are all carried on RBP4. So what are you sending out of your liver? Right? And also, I'm not really sure. Well, we can store it on cellular retinol binding protein. So in your liver, you have cellular retinol binding protein. So do you have your AR there? You can't store AR in a stellate cell because it doesn't have the hydroxyl group, is my theory on that, because that's how you get it into stellate cells is with LRAT. That's why I think alcoholics don't have any vitamin A in their liver. It's because alcoholics make anhydroretinol. So the problem we have here is we don't know what's in our liver. And we can't assume that a high serum vitamin A indicates that we have excess retinol. I'll explain that more. So this graph over here is showing something important. Let me make sure I'm on the right track. I'll come back to this graph. Okay, so how do you know if you have high stores of vitamin A in your liver? Well, one way to check that is to look at your retinol esters. So you can ask your physician to order a serum retinol level and then also a retinol palmitate or retinol ester level. And if your retinol esters are more than 7.5% of your total serum vitamin A, that means that you have vitamin A toxicity in your liver. And so I calculated Zoe's and it's like 4.7. So she doesn't have excess stores of vitamin A in her liver, but her serum vitamin A has just now come down finally. So what is going on? She's never had a massive amount of vitamin A supplements in her life, but she had this very high serum vitamin A level. And so definitely get that checked out. You don't want to assume that you have a chock full liver full of vitamin A. Now, if you were taking high dose vitamin A every single day of your life or eating liver every single day of your life, then, it's more of a chance that you have a problem. But so what was going on with her vitamin A levels? So this is in February 2021. This is when I had started to give her plantain flour around this time. And you can see that her vitamin A levels creeped up. And I think that it was because she wasn't metabolizing vitamin A right. She was not able to go from retinol to retinol aldehyde, but even more importantly, she wasn't metabolizing alcohol right. And that led to her inability to get vitamin A out of the blood and into the cell, and it made her push the vitamin A from the peripheral cells back out into the blood. So, I'll show you again in a minute, but let's talk about the immune cells real quick. And because if you're telling me that it's fine that you have the vitamin A coming from your liver, well, the cells that are going to use your vitamin A from your liver, if they're going to access it from RBP4, they need to have STRA6, which is the pore that RBP4 connects with to drop the vitamin A off. So these are the cells of the immune system that actually have STRA6. And let's talk about that. So dendritic cells, which are the cells that produce the retinoic acid to hand it off to the T cells and B cells that are in the gut that need to do the work, it does not have STRA6. So it cannot get its retinoic acid on its own to make it differentiate into the type of cell that can produce retinoic acid. So it doesn't have STRA6. So your circulating RBP4 version of vitamin A is not going to be helpful for your dendritic cells. Macrophages do have STRA6. So that's helpful, right? But that ends up I think being your number one protector when you're on a low vitamin A diet is you get stuck with macrophages doing the work. T cells and B cells, they do have STRA6, but again, they don't have the machinery to be able to make retinoic acid. They get it from dendritic cells. So you end up overemphasizing macrophages as your primary source of immune function, and your macrophages, they actually come from monocytes. So monocytes to macrophage conversion does require some retinoic acid. So I'm hoping that, everyone is producing some. You can actually look at your CBC, and if you have high monocytes, then you're doing a not good job at making macrophages. So that's kind of an interesting thing. So yeah, macrophages have STRA6, dendritic cells don't. So what is STRA6? STRA6, I said, was a pore that's in our cells, and we need it to be able to get vitamin A off of RBP4. So hollow RBP actually contains, it's so opposite. It sounds like it's hollow, but hollow RBP actually has the vitamin A in it, and apo-RBP does not have vitamin A in it. It's the same exact molecule. It's just you call it hollow when it has it and apo when it doesn't have vitamin A. And so hollow RBP, I'm going to talk about what I think is happening with the, yeah. Oh, sorry. Hold on. I got to pause. I got to collect my thoughts. I've been talking a long time. Okay. So hollow RPB, it has the vitamin A on it. And once it releases its vitamin A, it tends to just, go away. And then the apo-RPB without the vitamin A tends to stay associated with the STRA6 so that you can get the vitamin A in excess out of the cells. But high intracellular calcium will actually cause a cell to efflux more vitamin A back into the blood than if the intracellular calcium was normal. So it's intracellular calcium that is controlling whether or not you have your good, healthy vitamin A inside of your cell. And so I think what was happening with Zoe was she had calcium dysregulation, and also several of my other clients, their serum A is really high, and it comes down to calcium dysregulation. And actually in people with autism, they know that calcium is dysregulated. And so that's one of the finding, what would it be, like pathophysiology things that's going on with autism is there's calcium dysregulation. So Zoe has classic calcium dysregulation, but why is one of my big questions, right? Why is she pushing her vitamin A back out into the blood? She's not storing it in her liver. She's just not using it, and it's going back into the blood. We don't want it just staying in the blood, because remember, if your RBP4 goes up in the blood, it causes oxidative stress, because it actually works with those macrophages and turns on the enzymes that cause inflammation. Turns on the genes that cause the inflammation and high nitric oxide levels. Okay, so this is what I think is happening. I think that Zoe is struggling with a cytoskeletal protein, and also my other client Oasis, he helped me find this pathway. It's he is vulnerable to F-actin, and he has a problem with a capping protein called CAPZB, and he has severe high vitamin A levels with a peripheral deficiency, including like deficiency within the bones, not able to make platelets. It's pretty severe because he struggles with keeping his F-actin capped in this, making a long stick. When you have these long sticks, then you actually have so it's F-actin. Sorry, I'm really am tired. Okay, let me try again. So F-actin, and becomes like short chains of G-actin. So these little balls down here, these little balls are actually G-actin, and then you have F-actin, which is a full long stick, and then in between you have short actin oligomers. And you have to be able to export calcium out of a cell, you have to go from F-actin to G-actin. Oligomers, they have to be short acting oligomers for you to be able to export calcium from a cell. If these are completely destroyed, you destroy your ability to regulate a major calcium pump called PMCA. And if your calcium pump is down, then your cytosolic calcium levels go up. That overactivates calmodulin. So calmodulin is what is saturated with calcium that causes the Strontics spore to prefer to export vitamin A back out onto ApoRBP. So if you happen to have destroyed F-actin, then you're no longer working your PMCA pump. Then you're reliant on a backup pump that's sodium dependent. And you're still able to get calcium out of the cells, but it's sodium dependent. And that's a struggle for many people, especially the majority of people that I've been working with. They have some type of metabolic acidosis or electrolyte dysregulation, and they're just struggling in general. So they have high cytosolic calcium. So what is causing this to happen? Well, let me see if let me go back to this one. I kind of had this slide. So what's happening is that F-actin is destroyed by anhydroretinol. So that's what this chart shows. So right here we have the time in hours, and then we have the percent of dead cells. Like, it actually destroys cells. Anhydroretinol can cause a cell to go through apoptosis because it destroys the F-actin cytoskeleton. So we have the time along the bottom. We have the number of destroyed cells. And you can see by 5 hours, the cells are completely destroyed. And then along this side, we have the decrease in F-actin content. And so it basically, as the time goes on and the cells are exposed to anhydroretinol, the cytoskeleton breaks apart. Okay. Let me see. I actually think I have a picture of that too. So let's go to this study because it's talks specifically about the immune cells that are so important for the gut. So, and the ones that, require retinoic acid, like you need your B lymphocytes, you need your T lymphocytes to be involved in the immune process. So anhydroretinol is a natural occurring inhibitor of lymphocyte physiology. And this is done, again, by Dr. Hammerling. I am going to send him an email and tell him thank you because he is helping my daughter and my other clients. So what he found in this study was that anhydroretinol, which is a natural derivative of retinol, and you find it in B lymphocytes, anhydroretinol reversibly it can be reversed. This is a positive thing. It reversibly inhibits retinol and a healthy other version of vitamin A called 14R HRR dependent effects. And it blocks, when you have anhydroretinol in the cells, it blocks B lymphocyte proliferation, so you can't make more, and it blocks the activation of resting T lymphocytes. So you don't have your T lymphocytes, they're not mobilized, they're done. So anhydroretinol, in the absence of alcohol, becomes rehydroretinol. So remember that diagram, by 6 hours, the cell was dead. But if you can get the alcohol metabolized out of your epithelial tissues and not have anhydroretinol, then you can save your immune cells. So alcohol metabolism is crucial to get your gut health under control because it's the alcohol that's causing the anhydroretinol, which is causing the immune dysfunction. Anhydroretinol can actually, in the absence of alcohol, be converted to 14-HRR. So that's important to know. We just need to get rid of the freaking alcohol. So this is a really neat picture from the retroretinoids and regulated cell growth and death. And it shows the T cells. So the T cells, 3 hours after exposure to anhydroretinol, the cells had lost their ruffled appearance. They exhibited numerous blebs. I guess a bleb is this. They looked bloated and in part took on a cratered appearance. See all these little craters? And began to leak amorphous material. That's crazy. That's from anhydroretinol. So it's explains the damage and the poor immune function of my sweet daughter and many of my clients. They have the inability to control the alcohol levels in their gut for multiple reasons, possibly oxalate poisoning, like maybe on a high oxalate tube feeding, having sulfate metabolism problems. They can't make SULT1A1. Perhaps they have kynurenine pathway problems. They're not finishing it. They're not making de novo NAD. Anybody who's struggling with cortisol levels is going to struggle with that pathway, TDO pathway that's in the liver. But IDO is in the gut. And so nattokinase can inhibit the production of NAD in the gut. It actually inhibits the IDO enzyme. So very interesting. All right. And I put this slide again. So remember, if you destroy your F-actin, the cells will explode. But if you're destroying it, but also rebuilding it by having healthy retinol, you could still be struggling on and off within those first 1 or 2 hours of the alcohol being in the cell. You could be struggling with calcium dysregulation. If your calcium levels go high in a cell, and also magnesium is involved too, because calmodulin has to be loaded with calcium. If there's not magnesium on the EF hand, then calmodulin will stay on. And then that will actually cause the retinol to efflux from the cell back onto RBP4, and you have your high serum retinol that is not always a marker of a full liver. It could be a marker of calcium dysregulation. So again, if your calmodulin is staying on because of not enough magnesium, but also because your calcium pumps are destroyed, or maybe you're uptaking too much calcium, what would cause that? Acidosis. When you're in acidosis, you have more ionized calcium, and also your voltage-gated calcium channels are upregulated, and it pushes more calcium into the cells. So you have to really have active calcium exporters. So you have to have your PMCA pump active. You can't be making anhydroretinol. You need your sodium calcium transporter active. And that's crucial because you want your retinol to be inside of the cell, not exported out of the cell. So in a low dietary vitamin A state, where you're not eating vitamin A, the only cells that are going to be available to work in the gut are macrophages. But your macrophages, they're not going to be able to switch quickly from that anti-inflammatory state to an inflammatory state to recruit your natural killer cells. So you have to have vitamin A for that. If your RBP4 is delivering anhydroretinol, then retinoic acid is not going to be available. So even though macrophages have a STRA6, they express STRA6, if you're bringing anhydroretinol back to a macrophage, it's crap. It's not going to be helpful at all. Okay, and then if the cells already contain anhydroretinol, they may not be able to uptake retinol due to calcium dysregulation. So that's another factor. And due to constant insult to the gut from lack of B and T cell activities, sorry, that should be activation, macrophages become inflammatory, right? And that is not good, inflammation will cause leaky gut, and then LPS leaks into the blood and triggers more macrophages to take on immune function. So now you're not just dealing with your gut microbiome and trying to work your immune system in there. Now your entire body is trying to fight off the effects of LPS. The LPS is in your blood, it's in your liver, it's infiltrating into the body. So having no vitamin A in your diet or being a maker of anhydroretinol is destroying your immune function and leading to LPS. I could have probably done a better job on the clicking. So what is the solution? Well, you get good at alcohol metabolism and you eat some vitamin A, right? Eat some vitamin A. Get good at alcohol metabolism. That's the key. And actually get good at calcium regulation as well. So, and I'll work on more videos to try to help people out because it's wonderful. I'm getting my sweet daughter out of this mess and a couple other clients. It's beautiful. Okay. So whether you're avoiding vitamin A or being bad at making retinoic acid or you're high in anhydroretinol, the immune system is suffering in the gut, resulting in excess LPS, and it's getting into the body, right? And that's making you have to use your macrophages. The macrophages are in an inflammatory state because they've lost their ability to switch. They can't switch back and forth. So lack of adequate vitamin A in the body can contribute to the lack of dendritic cells being produced, resulting in poor immune coordination too. I think that I probably didn't need this repeat slide. Excuse me for being sleepy. And then what else was I going to say? Oh, macrophage production could also be reduced due to monocyte to macrophage conversion is induced by retinoic acid. So you could be struggling with fighting off all of these pathogens. So more body cells may take on the itaconate pathway to help act as microbial agents to fight the war on invading bacteria. Okay, so now I want to explain the itaconate pathway and why. Why would this happen? Well, itaconate is antimicrobial itself. It inhibits isocitrate lyase in bacteria, which acts like an antimicrobial agent. So your macrophage, this is a macrophage, when it responds to LPS, it switches metabolism and it uses a component of the TCA cycle called itaconate. And the LPS triggers the production of ACOD or IRG1, an enzyme that converts itaconate into itaconate. And then itaconate has downstream regulatory effects on other genes. It will activate KEAP1. And it actually inhibits KEAP1, and KEAP1 is what holds on to NRF2. And so it will go, NRF2 will go, or NRF2, some people call it NRF2, will go to the nucleus and it will induce gene transcription for enzymes that help with cleaning up the inflammation, because your macrophages are causing inflammation on purpose to kill off bacteria. When they're confronted with this LPS, they produce hydrogen peroxide and nitric oxide. And so then the itaconate, which itself is antibacterial, will push the cells to make things to deal with cleaning that up, such as glutathione peroxidase, superoxide dismutase. But one of the other things that NRF2 does is it actually induces the genes for keratin production. And so keratin is so interesting because keratinization or keratosis pilaris, well, that has always been classically a sign of vitamin A deficiency. What if it's a sign of anhydroretinol toxicity? Now, keratosis pilaris has been associated with vitamin A toxicity too, but it's when we find high serum vitamin A in the blood. What if it's actually anhydroretinol toxicity causing the overactivation of NRF2? Here's another way it could do it. The scaffolding for KEAP1 to hold NRF2 in the cytosol and not have it go to the nucleus, the scaffold is F-actin. It's the cytoskeleton protein. So KEAP1 is held into place by F-actin. If anhydroretinol comes along and destroys F-actin, then KEAP1 is loose, and KEAP1 can't do its job, and NRF2 will go to the nucleus, perhaps. Pretty interesting, right? So here's a hypothesis. If the adaptive immune system is offline in the gut, we don't have our dendritic cells working, we don't have our B lymphocytes, we don't have our plasma cells, we don't have our T lymphocytes, then the whole body might join in to fight off the microbes. And this process is what causes hyperoxaluria. So here's the itaconate pathway. I don't have pyruvate on here, but we go from pyruvate to acetyl-CoA. And acetyl-CoA becomes citrate, and then itaconate, and then the enzyme IRG1, or so I can't remember the name for this. Sorry, I'm kind of blanking out. But basically, this enzyme makes itaconate. Itaconate becomes itaconyl-CoA, and that itself, if it's not metabolized away, will poison B12. And that causes low levels of SAM production, and that can contribute to poor methylation. And then itaconate will also inhibit succinate dehydrogenase, which will have an accumulation of succinate. An accumulation of succinate will turn off pyruvate dehydrogenase complex. And this cycle continues, and it can produce acetyl-CoA and pyruvate. The acetyl-CoA would likely become malonyl-CoA, so that would contribute to myelination of pyruvate dehydrogenase complex, and that would also lead to turning off of pyruvate dehydrogenase complex, and then the excess pyruvate builds up, and it can go to the peroxisome. So there's that. This is interesting. This enzyme, itaconyl-CoA hydratase, and I think I actually misspelled it a little bit. It is also the same enzyme as methylglutaconyl-CoA hydratase and enoyl-CoA hydratase. It's the same enzyme. It's moonlighting. So on an organic acid test, you could have high 3-methylglutaric acid, and that's just because you're tying up this enzyme dealing with the itaconate pathway. You could, but you likely don't have an inborn error of metabolism. You might have a SNP in this enzyme. It's also called AUH. You might have a SNP in this enzyme, making you more vulnerable. But something interesting is that if you have a buildup of itaconyl-CoA, it will actually inhibit heme synthesis in red blood cells, not heme synthesis for heme enzymes, but just red blood cells. And so if you have that, if you have low red blood cell count, that's a sign that you're stuck in the itaconate pathway. If you, it's like anemia of chronic disease. That's where it comes from. And it poisons your B12 too, so that's potential cause as well. Oh, but I should say the cause of the low heme production is this itaconyl-CoA inhibits the enzyme ALAS, so the first enzyme in heme synthesis. So the succinate buildup from inhibition of succinate dehydrogenase, that induces hypoxia inducible factor 1 alpha. So here's the electron transport chain. Itaconate inhibits this enzyme right here, succinate dehydrogenase, and then that triggers hypoxia inducible factor 1 alpha. And so this is quite interesting. So we've already kind of talked about succinate will inhibit pyruvate dehydrogenase complex, and that causes a buildup of pyruvate, and then this itaconate pathway can contribute to excess pyruvate as well. So the hypoxia-induced factor 1 alpha, it also has insane effects. So it will inhibit pyruvate dehydrogenase complex, and so that causes a buildup of pyruvate. It will also increase the pentose phosphate shunt pathway, which can put an additional burden on thymin But also, if you're already thiamine deficient, you'll have an increase in ribose 5 phosphate, and then that is pushed into the uric acid metabolism pathway, and you will have increased uric acid levels. And that will actually cause you to use more cofactor for metabolizing uric acid, the xanthine that's being produced and becomes uric acid. And so then you'll struggle with sulfate metabolism and get worse at alcohol metabolism. It's a lovely, nasty cycle. This hypoxia inducible factor 1 alpha is also induced by low oxygen levels. So hypoxia, so it could even be just poor skin perfusion, like Raynaud's, or it could be someone with asthma. It could be someone with sleep apnea. It could be low blood pressure because that causes poor skin perfusion, and that'll trigger this HIF1 alpha. So you could have a block at pyruvate dehydrogenase complex that is separate from gut dysbiosis. It can come from low oxygen levels. It also increases GLUT1 and it increases glycolysis, and you'll end up with more lactate. And again, lactate can go to pyruvate. So hopefully you're going to be able to pee it out, but you might be able to get some lactate. And there's strips on the market that are lactate, acid, and uric acid, like your pH. So it would be lactate, pH, and uric acid. And you could check those. And, if you don't, and it has ketones. So if you're not in ketosis, but you have a low urine pH and you have high uric acid, that might mean you're in lactic acidosis and you're pushing the pentose phosphate shunt pathway. So, hindsight, in my family, we switched to using gluconate versions of electrolytes because I knew that we were needing the pentose phosphate shunt pathway because everyone was in oxidative stress. But by doing that, I gave everyone high uric acid levels. Hooray! The problem is that uric acid also will induce this HIF1 alpha, and uric acid can be toxic to the liver. So not a good situation. Avoid gluconate versions of electrolytes if you think that you're stuck in this pathway. Okay, so a block in PDHC from either cestinylation. I didn't talk about glutarylation, but I will have to do another video on that. But it also comes from the itaconate pathway or hypoxia-induced factor 1 alpha. It explains the high level of pyruvate and glycerate, right? The glycerate is because too much pyruvate, and it explains the oxalate too, right? We figured out that's what's happening. But what is causing the increased glycolate? What's causing this increased glycolate levels on oat tests? Well, the factor hypoxia induces, sorry, I should just call it HIF1 alpha, H1F. I can't say it like that because it's HIF1 alpha. Anyways, hypoxia inducible factor 1 alpha, it actually has hydroxyproline. See these prolines? They become hydroxyproline. And when it's hydroxyproline, then it goes through polyubiquination and it's degraded, and the hydroxyproline is released from this factor that's being overproduced. So when you need extra hydroxyproline, you'll start to break down your collagen because that's where you can get the proline from. And so if your joints hurt and you're starting to have loose joints, perhaps like me, where my jaw will dislocate or my hip will dislocate, and I've been diagnosed with Ehlers-Danlos hypermobility, but I don't have the genes for EDS, perhaps it's because my whole life I've been stuck in this pathway. And so I've been breaking down my proline to make hydroxyproline. And then when I'm done using my hypoxia induced factor 1 alpha, I degrade it. Okay, and so here is hydroxyproline, and it goes through this pathway. And then eventually it comes out and it becomes glyoxylate and pyruvate, another source of pyruvate. And then if you have plenty of B6, your glyoxylate becomes glycine. But if you do not have plenty of B6, or even if you do have plenty of B6, I don't think you can like just push it one direction. You would use GRHPR to make glyoxylate into glycolate. And that is where the high glycolate levels are coming from. You're either hypoxic or you're stuck in the itaconate pathway and your succinate levels have increased, and that's inducing this hypoxia inducible factor 1 alpha. And that's leading to the breakdown of it and this excess hydroxyproline that needs to be catabolized. Okay, so now we're going to look at some this is Zoe's labs. So this is Zoe when she was 2 years old, and I took her to a psychiatrist, and because I wanted to figure out if her autism was just a genetic thing or if there was something else going on. And it was one of the friends in our support group for her syndrome who recommended this particular psychiatrist, and he actually diagnosed me with an autoimmune disease while I was in his office. It was really interesting. So she has all these crazy markers. She even had a marker for this tartaric acid is for aspergillus. And she does not have that marker anymore. And that is comforting because she was able to recover from that without having to have intervention. Or it could have been like maybe one of the other bacteria or something makes it, but usually it's aspergillus marker. So I'll have to look into that. Okay, so anyway, she had very, very high candida markers. Check this out. Her arabinose, 746. This girl was just a walking fungus. She had had many antibiotics because she had had a few surgeries, and she just couldn't catch a break. And so I think she was a maker of anhydroretinol. Look at her succinate. It was at 30. That's crazy high. So this is back when it was Great Plains and they had things in a different order. It's pretty interesting. So here is her succinate, super high. I think that's because she's stuck in the itaconate pathway. Here again is her oxalate level. Her glycolic acid is elevated. That is coming from hypoxia inducible factor 1 alpha, and her glycerate is coming from the combination of pyruvate and serine, making hydroxypyruvate and then being metabolized to glyceric acid or glycerate. And she wasn't finishing the kynurenine pathway. You can see she has really high quinolinic and kynurenic acid, and she was actually dropping biopterin into her urine, and that is because both quinolinic acid and kynuric acid will prevent the de novo synthesis of BH4, the cofactor needed for normal coupling of nitric oxide synthase. And so when you don't have enough BH4, you make peroxynitrite. So lots going on when she was little. She was a very sick baby. Let's see. So on this, she has high sebacic acid. That is actually it's supposedly a marker for short chain fatty acid deficiency, but I actually think it's a counter-regulatory effect of the body. It makes sebacic acid to downregulate the effects of LPS. She has really high ethylmalonic acid levels, like at 17. That's really high, and also methylsuccinic acid. And this is because of the itaconate pathway. Itaconate actually inhibits, the malonyl CoA. I can't remember the name of it. It's abbreviated MUT, M-U-T. It's B12 dependent, and it helps you get rid of methylmalonyl CoA so that you don't make methylmalonic acid. You can see that she's slightly on the higher end of methylmalonic acid. She doesn't have a B12 deficiency though. She's just blocked at that enzyme by itaconate. And what that does is it ties up some other enzymes And it results, and I'll do a actual video on this, but it results in increasing levels of ethyl malonic acid. Well, ethyl malonic acid will block neurotransmitter. It's neurotoxic to the brain, and it causes sleep apnea, and it causes hypoxia. So that's really interesting. And so she had a lot of that hypoxia inducible factor 1 alpha. So this is Zoe now. I'm so proud. I just want to be like, ooh. She actually in August last year, her 4-hydroxyphenylacetic acid, and it's not on this. I didn't get an oat, but I got it at a Quest. It was 98. She had strep, and it was like off the charts. I still think she suffers from SIBO. But barely. She barely has high levels. We've been working a lot on alcohol metabolism. That's what we've been working on. And her immune system in her gut is coming back on board, and she's making secretory IgA. And now we're going to work on gut motility, because that is very much related to calcium dysregulation. If you have too much calcium inside of the motor, what is it called? Migrating motor complex. I believe that it shuts off. That's my theory. I'm still working on that, but we're working on restarting the migrating motor complex so that we can get the bacteria out of her gut. We did a stool test, and she has normal stool tests. Like, she doesn't have anything really bad growing in there. Pretty cool. So then let's look at her oxalic acid. It's come down, but it's still above the normal level, and her glycolic acid is very high. And this tells me Zoe's major problem now is hypoxia, and she still has sleep apnea, and she's almost 13, and she's so ready to be in charge of her own life, and she will not wear her CPAP mask. And so she's hypoxic every night. She does not drop into the 70s. She drops into the high 80s. But if you're always in being hypoxic, then you're going to end up with high glycolate levels, and you're going to end up with problems with pyruvate dehydrogenase complex. But you can see she's doing a lot better overall. Her pyruvate isn't sky high. She's able to go from pyruvate to lactate, but that's because her lactate dehydrogenase is upregulated by the hypoxia inducible factor 1 alpha. So one very sad side effect for Zoe is she has chronic lower back pain because she has degenerative changes in her spine, most likely because she's not good at making collagen matrix for bones, because she's breaking down her proline to be used to make the use the proline on the hypoxia inducible factor 1 alpha. It's really sad. I'm actually considering adding more proline to her diet, but I'm pretty sure that's just going to make her glycolic acid go higher. So I'm not sure what I'm going to do there. I'm hoping to get her sleep apnea solved. And then her ethyl malonic acid has come down considerably. I do know that now that I just figured this out within the past year, that she has lower levels of mRNA for very long chain acetyl-CoA dehydrogenase, which actually can make it harder for you to deal with fatty acid metabolism, and it can lead to problems with ethyl malonic acid. So she's vulnerable there, but I don't think that was the cause of it being high and considerably compared to other people, because I've seen this on other people's oats as well. So there is the fact that malination, let's see, is her malinate on here? I don't know if I took a screenshot of it. No, I didn't take a screenshot of it, but her malinate was still a little bit high. And so it could be that she's just struggling with fatty acid catabolism in general, which, is an issue. Ethyl malonic acid can come from butyrate. And I used to be fearful of giving Zoe butyrate, but now that I know that, it's on one of my slides, but I didn't say it out loud, but butyrate can help the dendritic cells use retinoic acid. So I'm not going to be fearful about fiber for her. I'm just going to work on alcohol metabolism and on hypoxia. That's my goal. So this is one of my friends. And this is her oat. And you can see that she has a lot of gut dysbiosis as well. And this is her oxalate level. I actually think I already showed this particular set of labs, but you can see that her Itaconate is pretty low. Her citrate is pretty low too. I actually think that she has I think she might have been on a ketogenic diet or a low carb diet at this time. And so, overall, she has less going into the TCA cycle. But she still has high succinate levels, and that could be because there are two blocks going on there. I mean, it's not super high, but it's above the mean. I mean, it's in the average, but it's above, on this higher side. So she has the block from the itaconate pathway potentially at succinate dehydrogenase, and then itaconate blocks methylmalonyl CoA also. So all of the branched chain amino acids like valine, I can't isoleucine. I think it's just valine and isoleucine. But again, I'll do a video on it. Those are pushing into it's valine, 309, isoleucine, and methionine go through propionyl CoA and then to methylmalonyl CoA and then to succinate. So all of those are hard for her to use because there's a block potentially at MUT, the enzyme that converts methylmalonyl CoA to succinyl CoA. It can be blocked by itaconate. So the succinate is likely coming from the GABA shunt, but then there's a block at succinate dehydrogenase, so it's increased right here. Anyway, turns off pyruvate dehydrogenase complex, and then the pyruvate is pushed into the peroxisome and causes high oxalate. Oh, another thing that could be happening if you look down here, all her glutaric acid derivatives are high, and so she could be in the excessively making glutaric acid, and that is causing glutarylation, and that is actually induced by hypoxia inducible factor 1 alpha. Also, it will inhibit the enzymes involved in that pathway, and it will lead to increased glutaric acid levels. So you can see here, her glutaric acid is high, and that can be a marker of a B2 deficiency, but I'm thinking it can also be a marker of the itaconate pathway. There's a lot going on here, so I potentially might kind of wrap my brain around it and see if I can do a separate video about these labs. So another proof with this particular friend that she's struggling with pyruvate and serine becoming hydroxypyruvate is that her serine is low. So she is pushing pyruvate to the peroxisome and combining it with serine and making hydroxypyruvate. And so here's her serine getting into the peroxisome, combining with the excess pyruvate, making hydroxypyruvate, tying up GR-HPR and making glycerate. Let's go back to her glycerate. Here's her glycerate. Her glycolic acid is low, so maybe a little bit less of that hypoxia inducible factor 1 alpha. Mostly she's probably dealing with that excess glutarylation. Also, she has high uracil, which can be converted to, it can be also converted to pyruvate, but it also can be converted to malonyl CoA. I don't have it on here. Darn it. I didn't put all the screenshots, but it's pretty interesting, right? So, again, this could be a B2 deficiency, or it could be a product of immune function. And you can see she actually has a normal F-malonic acid, and that's kind of encouraging. I kind of want to look at what the difference is between her and my sweet girl Zoe. So there's her serine. So serine and pyruvate make hydroxypyruvate, which is metabolized to deglycerate, and then that ties up GR-HPR, and it's not going to metabolize glyoxylate to glycolate, and you're going to make oxalate, and her glycolate is pretty low. Um, oh, this is me. These are my little notes. So these are notes that I wrote, but I didn't finish writing them on the slide. Sometimes I forget to finish things. So this is actually my oat. I was feeling really bad, like really, really bad, and I decided to do an oat on myself, and I do not have hyperoxaluria. Um, I I do have low pyruvate and I have high lactic acid. I believe that I'm pushing my pyruvate into malate because I think that I have a functional B6 deficiency. I do take betaine, but it doesn't seem to either. It's not working because I have a ton of lipid peroxides going on right now because I have copper deficiency. So my ALDH791 is really busy in the mitochondrial membrane. So I think I have a functional B6 deficiency, and I can explain why in a second. But so I think I have a high I am stuck in the Itaconate pathway. Maybe I actually kind of think that all of my citrate is cytosolic, and I'm actually not making much mitochondrial citrate, and that might be some of the problem. I think that I'm actually having hypoxic episodes. The reason why I think that is because my ethylmalonic acid is high as well. So I'm still trying to work out my own labs. I got a bit of brain fog. So I'll get back to it. The reason why I think I have a functional B6 deficiency is because my B6 marker here, which is pyridoxic acid, is low on the low side. My blood B6 is high. My zinc levels are marginal. My alkaline phosphatase is low. I'm struggling with B6. I think that I'm also making P6C because I'm so busy with lipid peroxides. So I'm thankful that my brain is still working. I make my family laugh because I said all I'm going to be doing is thinking. I'm not doing housework. I'm not cleaning. I'm not making food. I'm trying to preserve brain function. So I hope this video is helpful to people. I actually think that my PDHC is turned off from malonic acid, from the citrate being in the cytosol and making because I have a B6 deficiency. I'm not finishing GABA shunt. I'm not pushing anything back into alpha ketoglutarate. Pretty interesting. And that is me. I actually have this. This is a sign of high ethylmalonic acid. I will stand up and my feet turn blue, and I have postural orthostatic tachycardia syndrome. So what if EDS in POTS is just overactive Itaconate pathway and hypoxia inducible factor alpha 1? Probably should have said that at the beginning of the video, right? Pretty interesting. I've had this going on for a while. So one more thought about this before I finish. I thought that retinol aldehyde was stealing Zoe's ethanolamine, but what if it's not that? I mean, it's possible she's making lipofuscins. Anything's possible. That's like a new thing. Anything can cause anything is what one of my friends is saying, and I love it. What about low ethanolamine levels on a plasma amino acid test? Is it from A2E formation, or is it possibly from hyperoxaluria pathway too? Because if you're pushing your serine into making hydroxypyruvate to deal with your excess pyruvate, then you're not having enough serine to basically access the ethanolamine from phosphatidylethylamine. So phosphatidylserine synthase uses serine to mobilize ethanolamine from phosphatidylethylamine. So it could just be lack of metabolism in the plasmalogen pathway. And that means that there's less ethanolamine to make the specific CDP ethanolamine. So then that causes problems with neurotransmitter homeostasis, synaptic membranes, membrane vesicles, antioxidant, plasmalogens are actually antioxidants, but lots to consider there. A whole new way of thinking of things. So signs of being in the Itaconate hypoxia inducible factor 1 alpha pathway, and blocks on at PDHC include high glycolate or glycerate levels, high oxalate levels, low serine levels, low coenzyme Q10. It is possible to have a high coenzyme Q10 on a lab because perhaps when you start to break down, like keep um, like if you are making coenzyme Q10, but you're doing a lot of NRF2 pathway, then you could have a lot of coenzyme Q10 or ubiquinol kind of breaking off of that pathway. So I don't know if low or high is necessarily. It's probably one or the other. In between, you're probably okay. High or low pyruvate or lactate. High uric acid means you're pushing the pentose phosphate shunt pathway. Low uric acid doesn't mean you're saved. It just means you might have a MoCo deficiency, and then you could have high levels of xanthine. You're not making uric acid. You could have a normal anion gap acidosis without ketones. That would mean you're probably on lactic acidosis. You could have, if you're having ketones and glucose is normal and you're eating carbs, that could be a sign of anhydroretinol in your PKCdelta signalosome. You could have crusty elbows and knees and orange calluses, which, I thought, I must be just storing beta carotene in my orange callus. No, my whole hand would be orange. I really needed to think about that. I actually think that it's keratinization from that hypoxia induced factor 1 alpha causing, from the Itaconate pathway. Itaconate pathway is causing me to be overactive in NRF2. High ethylmalonic methylsuccinic acid. That's from blocks at succinate dehydrogenase, and it puts a burden on the propionyl-CoA carboxylase and acetyl-CoA carboxylase enzymes that also metabolize ethylmalonic acid. And then methylsuccinic acid is a breakdown of ethylmalonic acid. So any of these makes me think you might be stuck in the itaconate pathway. High glutaric, 3-methylglutaric acid, 3-hydroxyglutaric acid, 3-methylglutaconic acid. All of these are metabolized by itaconate-CoA hydratase, which is also known as AUH, and then that can cause an increase in these markers. So it doesn't necessarily mean that you have the genetic syndromes for these things when the markers are just a little bit high. It means that your immune system is overactivated. Potentially your entire body is joining in on the immune response, and that's altering your metabolism. And then high sebacic acid, it's made by the body, maybe potentially to block LPS, and there's like a study on that. So what are the solutions? A ketogenic diet might help temporarily if you're an oxalate maker. It might relieve some of the burden that you're having and make you feel a little bit better, but it could potentially alter your immune capacity because if you're in the setting of making AR, you're on a low vitamin A diet, and you're just struggling with your immune function, if you go ketogenic, you're reliant on gluconeogenesis to fuel your immune system because that's what it runs off of is glucose. And that's part of the reason why it does that metabolic switch is because the glucose actually can help T cells have better memory, and it helps fuel T cells to give them the energy to fight infections. Or you could just try a very low carb diet to see if it takes some of the burden away. That could be potentially helpful too. A low oxalate diet would be helpful because you got a lot already, and you don't want to inhibit your cytosolic NAD recycling in your gut because you want to eventually be using vitamin A in your immune system. I would get with a specialist to work on gut health to prevent the alcohol, producing species of crap that happens in our gut, like Candida, Aspergillus, Staphylococcus. I think I think E. coli makes lactate. I'd have to go back and look, but I do want to make another video about this. So work with a gut health specialist to prevent anhydroretinol formation, and also to lower your LPS, because if it's not anhydroretinol screwing you up, it's definitely excess LPS. You want to eliminate sleep apnea and hypoxia because, and this can be caused by high ethylmalonic acid or actually high GHB. So you could check an OAT to see if you have those. You might need to do CPAP. temporarily to stop the cycle of damage. It might be beneficial to be on it for a little bit until you can get out of this immune pathway. And then you want to troubleshoot ongoing oxidative stress. So restore minerals, zinc, copper, selenium, manganese, not magnesium. Although you probably are wasting magnesium in your urine because of acidosis. You want to make sure you get normal amounts of vitamin E, vitamin C, and coenzyme Q10 if you need it. You might not need coenzyme Q10. You might be able to make it. You make it from pantothenic acid. So replenish your B vitamins, thiamine, lipoate, pantothenic acid, riboflavin, and B6 if you have a true deficiency, and then potentially betaine for a functional deficiency would be good also. But just realize that when you restore B6, if you actually have a B6 deficiency, it might also restore you to making oxalate. So that's interesting. And then what if adverse reactions to thiamine might be a symptom of anhydroretinol toxicity? So if you take thiamine and then everything gets worse, maybe you have AR in your PKC delta signalosome. That's possible. One more thing about this is that biotin, I didn't put biotin here, but biotin and pantothenic acid actually compete for absorption. So be aware of that. And lipoate too. I wouldn't necessarily take lipoate. We don't incorporate it into our pyruvate dehydrogenase complex. We actually can just use it as an antioxidant when we take oral lipoic acid. I would just think about biotin. I struggle with biotin when I take it. It seems to make candida flare. Okay, so I think that's it. I'm getting a little brain foggy. And I want to do another video. It'll either be on unintentional ketosis, the mocosteel and the SOLT1A1 crisis leading to high alcohol levels, detecting immune dysfunction or hypoxia on oat, which I kind of already went through that stuff, but it needs a better sussing out. Or I might just make another video singing the poo-poo song. And so if you want to choose one of these options, you can put it in the comments. All right. Thanks for watching. I'm going to go take a nap now because I have strep and aspergillus and feeling really, really tired, but I know the way out, right? That's the good part. All right. Thanks.